In this clip we're going to start looking at Punnett squares, and in particular look at some things that you need to know when you're answering questions on Punnett squares. You can't get away from learning a few definitions, and often learning a definition and giving that an exam is pretty much what you need to do for achievement. A few definitions and one intelligent sentence and you may just fluke yourself an achievement. So genotype. We're going with the definition of a genotype as the two letters that represent the two alleles for a gene. So you could be homozygous for tongue rolling with the genotype big T, or not tongue rolling, little t. Homozygous means both alleles are the same. So big T, little t. You could be heterozygous for tongue rolling. And heterozygous means those two alleles are different, so little t. It's the alleles that determine the phenotype. Your phenotype is what you see in the organism because they have their genes. The features or the traits or the characteristics means the same thing, so it's the hair colour, the eye colour, the height. That's what phenotype is. So remember, one set of chromosomes in any organism comes from the egg and one from the sperm. So we've all got a double set of chromosomes in our genes, 2 copies of every gene. Usually when there are 2 different versions, alleles, of a gene, one will be dominant. If you've got one of each allele, the effect isn't usually blended or somewhere in the middle. It's one thing or the other. The allele that shows in the phenotype when one of each type is present is the dominant allele, and we give that one the capital letter. So down below we have a guinea pig that has a black allele in the middle and the white allele, but he appears black. Therefore we call black dominant and give that allele the capital letter. We can see to be a white guinea pig you have to have 2 copies of the little b allele, and the big B has to be absent. So learn these definitions of a dominant allele and a recessive allele. You're bound to be asked for one of them. Now let's think about how it all works. I'm using as an example dimples. Dimples, those little dips in your cheek, are caused by a dominant allele. I've chose to make my mum and dad here, they're actually just girlfriend and boyfriend, that's why they're smiling. I've chose to make them heterozygous. They have got 1 copy of the dominant allele, therefore they have dimples, and 1 copy of the recessive allele, which is no dimples. Remember, the gametes only have 1 set of chromosomes, so they only have 1 copy of each gene. In this case, a sperm will be carrying a big D or a little d, and the egg will be carrying a big D and a little d. So we can use Punnett squares once we know the parent's genotype to make some kind of prediction about the likelihood of them having children. So our Punnett square could predict the likelihood of getting this little chap with his dimples. Isn't he cute? And this is how we do it. Mum here is the heterozygote up here. Dad here, heterozygote over here. And then remember, 1 allele goes into 1 gamete, 1 allele into the other, and here are the gametes that we are showing here. And we then say, well, what would happen if this sperm met that egg, or this sperm met that egg? The one that it's circled gives you the likelihood of having a normal child, and they're delighted to report the likelihood of having a dimpled child is the other 3, all of those with a big D. Our phenotype ratio, the features we see in the children therefore, are 3 dimpled to 1 not dimpled. Never just write 3 to 1 or you'll get no marks, but you must write the feature following the letter. If they ask for genotype ratio, you can see there is 1 big D to 2 big D little d's to 1 little d. If they ask for a phenotype ratio, Do not give both the phenotype and genotype. They will assume you don't know what the word means and they won't give you the mark. Here's some more phenotype ratios. What if both parents were homozygous, had the same alleles, one for the dominant allele and one for the recessive? In this case, all the children would be dimpled. Or if one was a heterozygote, like our example, and the other was homozygous recessive, as in the two normal alleles. In this case, we'd have one dimpled to one non-dimpled. So those ratios are really useful to remember. If we look at this example now, red and white plants, where big R is the red allele and little r is the white, it's a good question to remember how those phenotype ratios are produced. So remember, if you had the double heterozygote, big R little r times big R little r, you saw a phenotype ratio of 3 to 1. So if you were given a question where it said, in the cross, I crossed some plants, and in my offspring I had 310 red plants and 98 white plants, you'll say to yourself, that's just about 300 to 100. That's pretty much close to 3 to 1. Therefore, I know that the parents were pretty most probably heterozygous, big R little r times big R little r. And you can do the same with the other ratios. If you got a question that said 134 red to 125 white, that's roughly 1 red to 1 white. The parents are most probably big R little r times little r little r. And if the results are all red, one of the parents at least are most probably big R, big R. You can't make these sort of predictions though if the sample size is really small. In the examples I've given you, I've chosen big numbers, so it's enough to kind of come close to the statistical prediction of a Punnett square. So if you get a question where they're asking why in a family, for example, do I not have 3 children with dimples and 1 child without, there are several ideas you need to cover in your answer. Firstly, you need to say a Punnett square is only a prediction. Fertilization is actually a random event, and it's down to chance which gametes are actually involved in fertilization any particular time. Families are generally a small sample size, so you can't actually expect the family ratios to reflect the prediction of a Punnett square. Just like if you tossed a coin twice, the prediction is there'll be 1 head and 1 tail, but you may not necessarily get that. You may get 100% heads, 2. But the more times you toss the coin, the more likely your ratio will come down to close to 1 to 1. So your last idea will be to get an actual result that matches a Punnett square, a lot more crosses would be needed, or a larger sample size. The next tip in this clip is if you get a pedigree diagram discussing a trait and it doesn't tell you if it's dominant or recessive, and that's part of what you have to do. Be careful that you don't pick a section in the Punnett square that you think shows dominance. Check. Could you also explain that result as recessive? Because if you can, then you can't be sure of what you are choosing. The best way to show a trait is dominant is by showing it can't be recessive. Because if it can't be recessive, it has to be dominant, and vice versa. There's 2 patterns to look for. One of them will show you that it can't be recessive, and the other will show that it can't be dominant, and now I'm going to explain them. This part of a pedigree chart cannot be showing a recessive trait, so it must be dominant. On the left-hand side, try and make it recessive. The disease sufferers are coloured in red, and if we want this to be recessive, we give them the recessive genotype, little d, little d. But their child does not have the condition. They are normal. That must be dominant. Therefore, they need a dominant allele, and the parents can't give them one, so this just doesn't make sense. So this condition cannot be recessive. Just check to make sure you can make it dominant, and yes, with 2 heterozygotes for the parents, we can. The second pattern to look for is one that means it cannot be a dominant trait, therefore it must be recessive. Can we make The one on the left, dominant. Well, there's the disease sufferer. We give them a capital R for dominant. But his parents, who don't have the disease, therefore, must be recessive, little r, little r. So they can't have a big R to pass on. So again, this explanation is nonsense. So let's check. Can we make it recessive? Yes, our child with disease, we make them recessive. The parents are dominant normal and heterozygous. So look for a part in your Punnett square that has either of these situations, as in this slide or the slide before, to tell you that it can't possibly be dominant, so it must be recessive, or it can't possibly recessive, so it must be dominant. And one last word about those slides. It's okay to say the disease is dominant because most of the people in the pedigree are showing the disease. Yes, include that idea, but also look for a patch of the pedigree that makes it definitive. It can't be recessive. Next question you often get is how, for example, how can a blue-eyed recessive trait child have two brown-eyed parents, which is the dominant trait? And I know you're all sticking your hands up yelling, they're heterozygous, they're heterozygous. Yes, yes, well, that's true, but nobody's going to give you a merit grade for saying two words. This is what you need to cover. You must say the blue-eyed child must be little b, little b. The parents have brown eyes, so they must have a big B allele. But since the child received one allele from each parent, the second allele in the parents must be little b. So the parents must be heterozygous, big B, little b. So if you want the grades, you just have to do the writing. Our next tip is if you had a question like this, where you had to figure out and explain, for example, a brown-eyed person's genotype. And let's say you realised they had to be big B, little b to have that blue-eyed child. So you're attempting to explain why the parent is big B, little b. But remember, you have to explain both alleles. You have to say, and this is the bit a lot of you miss out, you have to say they have a big B because they have brown eyes and brown is dominant. And then you can go ahead to say, and they have a little b because they had a blue-eyed child, which needs two little b alleles, and the child gets one from each parent. Again, to explain the big B, little b genotype, you need to say these four ideas. Question 7. If ever you get asked a question, family of 7 girls, what's the likelihood the next child will be a boy? It's always 50%. Every time an egg is fertilized by a sperm, there is a 50% chance the sperm could be carrying an X and a 50% chance the sperm would be carrying a Y. Don't get any more complicated than that. So, practice your Punnett square problems from anywhere. This is a new standard from last year, but the inheritance material doesn't change. The inheritance problems in the old genetic standard are just like the inheritance problems in this standard. Your Purple ESA book, old study guides, old NCA papers are perfectly fine for helping you answer these questions. Just remember to answer fully. When you're discussing it, always give genotype, phenotype. The boy has blue eyes, little b, little b. Using the letters of each subject wherever you can. State the obvious and show what you know. There'll be marks for saying stuff that you think is too obvious to write down. The next 3 slides speak for themselves. When we talk about Punnett squares, there was indeed a man called Mr. Punnett, and this is him here. So good luck with these. I notice when I'm looking at the views on my clips that hundreds are watching 4 of the 5 clips that are posted and very few are watching the ones called genes and proteins. I cannot stress strongly enough that the whole point of inheritance is inheriting a gene that allows you to produce a protein that has some effect on your body. There will be important stuff in that clip, so please go back and watch it. Good luck with your exams.